Welcome to Botany class, Chapter 2, Mineral Transport, Nitrogen Cycle. In this, Metabolism of Nitrogen, today's topic, and majorly we will discuss nitrogen cycle, how it happens in our nature. Nitrogen is a constituent of amino acids, proteins, hormones, chlorophylls, and many vitamins. Plants compete with microbes for the limited nitrogen that is available in soil. Nitrogen exists as two nitrogen atoms joined by a very strong triple covalent bond. This molecular nitrogen to ammonia or nitrogen oxides, nitrites, and nitrates is termed as nitrogen fixation. In nature, there is a three steps forming of nitrogen, while one is lightning, second ultraviolet radiation. These two convert NO, nitrogen, NO2 or NO3 by the process. One more is industrial combustions, forest fires, automobile exhaust, and power generating stations also source of atmospheric nitrogen oxides, but these are artificial process. Nitrates and ammonia formed or absorbed by plants and converted into amino acids, proteins, enzymes, nucleic acids, pigments, and hormones. So this is very important bit, choosing the choice from the question. When plants are eaten by the animals, this organic nitrogen is passed into animal body. The process of absorbing nitrates, ammonia, and chemically binding the nitrogen with other elements to produce organic nitrogen in plants and thereby into animals constitute nitrogen is called as assimilation. Decomposition of organic nitrogen of dead plants and animals into ammonia is called as ammonification. This ammonia volatiles and re-enters the atmosphere or nitrate by soil bacteria in the soil decomposition. Ammonia is first oxidized to nitrogen by the bacteria. Example, Nitrosomonas or Nitrococcus. Further oxidized to nitrate with the help of the bacterium Nitrobacter. These steps are called nitrification. Then these nitrification bacteria are called as chemoautotrophs. The nitrate thus formed is absorbed by plant and transported to the leaves. It reduces to ammonia, finally forms the amino group of amino acids. Then nitrate present in the soil is also reduced to nitrogen by the process of denitrification. So this denitrification is carried out by the bacteria Pseudomonas and Thiobacillus. Let's discuss biological nitrogen fixation. In this process, the reduction of nitrogen to ammonia by living organisms is called as a biological nitrogen fixation. Enzyme nitrogenase, which is capable of nitrogen reduction, is present exclusively in prokaryotes. Such microbes are called as N2 fixers. N triple bond N by the process of nitrogenase enzyme convert into NH3. Here the nitrogen fixing microbes can be free living or symbiotic. Free living nitrogen fixing aerobic microbes are Azotobacter and Bergerincia, while Rhodospirillum is an aerobic and anaerobic bacillus free living. A number of cyanobacteria such as Anabina and Nastak also free living nitrogen fixers. Let us discuss symbiotic nitrogen fixation. Several types of symbiotic nitrogen fixing associations are known. The most prominent among them is a legume bacteria relationship. Species of rod-shaped rhizobium, such as a relationship with the roots of several legumes, such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans, etc. Here, these common association on roots as a nodules. These nodules are small outgrowth on the roots. The microbes Frankia also produce nitrogen fixing nodules on the roots of non-leguminous plants, example Ulus. Both rhizobium and Frankia are free living in soil, but as a symbionts can fix atmospheric nitrogen. Here, nodule formation involves a sequence of multiple interactions between rhizobium and the roots of the host plant. The principal stages in the nodule formation are summarized. Rhizobia attracted by the sugars, amino acids released by the host legume, multiply and colonize the surroundings of roots and get attached to epidermal root hair cells. The root hair curl and the bacteria invade the root hair, an infection thread is produced. Carrying the bacteria into the cortex of the root, bacteria initiate nodule formation in the cortex of the root. Then, bacteria released from the thread into the cortical cells of the host stimulate the host cells to divide. Thus, leads to the differentiation of a specialized nitrogen fixing cells. The nodule thus formed establishes a direct vascular connection with the host for exchange of nutrition. So these events are depicted in general process. The nodule contain all the necessary biochemical components such as enzyme nitrogenase and like hemoglobin. Enzyme nitrogenase is a molybdenum iron protein and catalyzes the conversion of atmospheric nitrogen to ammonia. Here, the enzyme nitrogenase is highly sensitive to the molecular oxygen. It requires anaerobic condition. The nodules have adaptations that ensure the enzyme is protected from oxygen. To protect these enzymes, the nodule contain an oxygen scavenger called leg hemoglobin. It is microbes live aerobics under free living condition, but during nitrogen fixation events they adapt to anaerobic condition, thus protecting the nitrogenase enzyme. Here the nitrogenase require very high input of energy, 8 ATP for NH3 produced. Fate of ammonia. At physiological pH, the ammonia is protected to form NH4 ion. Then, later it is quite toxic to plants and hence cannot accumulate in them. So, in this, two main ways in which this can take place. One is reductive amination, second is transamination. Reductive amination process reacts with alpha ketoglutarate acid and forms glutamic acid and indicated in the equation. And then transamination involves the transfer of an amino group from an amino acid to keto group to keto acid. Glutamic acid is the main amino acid from the transfer of NH2. Here the enzyme which is transaminase catalyze all such reactions. Here most important amides asparagine and glutamine. In plants there is a structural part of proteins. Addition another amino group hydroxyl part of the acid is replaced by the NH2 radical. Since amides contain more nitrogen in their amino acids, transported to the other plants part via xylem vessels. Along with the transpiration stream, the nodules of same plants, soybean, export the fixed nitrogen as a urides. These compounds have high nitrogen to carbon